Electrical signals are used by neurons and muscle cells in animals. An electrical signal is a flow of ions. But how do cells create a flow of ions? You need a concentration gradient that's created by a pump, and you need to control the diffusion of ions using ligand-gated channels and voltage-gated channels. We saw this in plants, remember? We saw ligand-gated channels that are activated by glutamate. And glutamate is a ligand, or a chemical that binds with a receptor and opened a channel, and we saw calcium ions flow through that channel down calcium concentration gradients. We also saw in stomatal control hydrogen ion pumps that created a hydrogen ion concentration gradient. And this concentration gradient of hydrogen ions triggered voltage-sensitive potassium channels, and potassium flowed down its concentration gradient into a cell. These ligand and voltage-gated mechanisms are similar in animal cells. For example, we will see more details, but let's look at the basic mechanism. So in an animal cell, animal cells have sodium pumps, sodium-potassium pumps, but we're going to focus only on the sodium. And animal cells have ligand-gated channels. And in this case, we might have some kind of ligand like acetylcholine, for example, a neurotransmitter that binds to a receptor on a sodium channel, opens the channel, and permits sodium to enter an animal cell. Similarly, in this animal cell, may also have voltage-sensitive sodium-gated channels, and as these sodium ions, for example, build up, enter a cell, they may trigger this voltage-gated sodium channel and permit more sodium to flow into an animal cell. In addition, this kind of voltage channel, we may have a voltage channel sensitive to sodium ion, but perhaps this is a calcium channel. We're going to see a little bit more detail on how concentration gradients, ligand-gated channels, and voltage-gated channels for different ions are important in muscle cells and neurons communicating with each other.